Advancing for Colloquia series with Girvan. I don't know if you're the last speaker of the season, but you're certainly the only local speaker that we've had here.
There's another one.
With the, I'm sorry?
There's another one.
Oh, there's another one? Yeah. So you're not the last one. Just a little bit of information concerning who she is. She's with the physics department with a joint appointment with Higgs. If my memory serves me right, you got your PhD in physics from Cornell and had a postdoc with Mark Newman at Sanitary Institute.
Yes, actually, Mark was no longer there when I was a postdoc.
And in fact, she started her career with a bang, since she has an extremely well reference paper on community detection of some modularity techniques. And today, she will be our speaker, and I think no need to repeat the title. You have the floor.
Thank you. So it's a pleasure to be here in this seminar series that appeals to so many different audiences. So to just tell you a little bit more about myself, as you heard, I come from a statistical physics background, and I've worked on a lot of network problems, many just involving understanding the different structural properties that are important in complex networks and that are important to dynamical systems on complex networks. More recently, I've been applying some of this more fundamental theoretical work to biological applications, and that's what I'm going to talk to you about today. So in particular, I'm going to talk about looking at annotation data, gene annotation data, and doing a network analysis of this kind of data to gain insights into gene interactions and the organization of biological function. So gene annotation data. So basically you have lots of genes, for humans on the order of 20,000. And genes serve different functions, and biologists can submit annotations that say this gene does this function to the database, and what we're going to do is mine the network structure in those annotations. So before I begin, I would like to acknowledge my collaborators. This work was primarily done by Kimberly Glass, who was a graduate student with me who has now moved on to a postdoc in computational biology at the Dana-Farber Institute at Harvard. And also in collaboration with Ed Ott, who is a physics and ECE professor here, and Wolfgang Loser is another physics professor, who actually got us into this problem. So before I really talk about the biological problem, I know you've had a physicist or two at these talks, but sometimes it sounds strange to people when I say I'm a physicist and I'm working on this biological network problem. So I just want to sort of outline why statistical physicists are drawn to these kinds of network problems and sort of what we think we can bring. So statistical physics, as you may know, is well equipped to deal with networks that are highly regular. For example, the lattice of connections of atoms in a solid, or highly random. So if we consider interactions to be fully mixed, if we consider the molecules in a gas in the air, and we consider, okay, every atom has, on average, the same likelihood of interacting with every other atom, we can do what we call mean field models. So we're really good at these sort of dimensional models on lattices and the mean field models that assume perfect mixing. But the appeal of networks to us is that heterogeneous networks represent a new area in which to apply, to extend the tools of statistical physics for dealing with these many body problems. So physicists in general, and statistical physicists in general, we're really interested in solving the overall behavior of many body systems, and so networks represent a new kind of challenge and complication for that. And then in addition, often people think of physics as string theory, particle physics. That's the kind of physics you often hear about in the news. But statistical physicists have a long tradition of applying the mathematical approaches developed for this field to many body problems in other fields. And so some examples are, you can look at problems like animal flocking. It's sort of similar to spin glass problems. And you can look at market behaviors in similar ways with models from physics. So we kind of take this approach in looking at these network problems. So back to the problem at hand that I want to discuss today, why do we want to analyze this graph structure of gene annotations? Well, there are two reasons we want to do this. The first is to determine if there are undocumented biologically meaningful relationships between terms. So when I say terms, I mean biological functions, the terms that are associated with different biological functions. And so biologists have a conceptual view of how functions are related. But can we actually go through and mine the annotation data to find relationships between functions that are not yet known? And so that's one of our goals. But we can also turn this annotation data on its head and say, well, the annotation data also provides information about relationships between genes. So how can we begin to mine this data to understand large-scale functional relationships between genes? So these are the two parts of my talk. First, I'm going to say, okay, well, how can we relate terms and functions? And then how can we use the network to gain new insights in the relationships between genes? So the structure of the gene ontology. So as I said, annotation data it comes in a database where genes are annotated to functions or terms. But the gene ontology basically provides a controlled vocabulary for biologists to use when they make these annotations, because we don't want individuals to call two individuals to call the same function by different names. So this is a controlled vocabulary that's provided. And it's, it's been around for a number of years, and it's really quite well used in this annotation context. So the gene ontology, again, it's a hierarchical classification system for biological functions. And the hierarchy takes the form of a directed acyclic graph. So this is a general term, and the gene ontology is divided into three main branches, one of which is molecular function. And so this is then differentiated further into functions that are more specific, that are each differentiated further down. But you see, sometimes there's crosstalk between the levels of further differentiation. And so what happens here is that genes are assigned to terms or functions. And so, we might have this function chromatin binding, and when biologists submit annotations, they'll submit an annotation of a specific gene to a function. And these are done for different species. And the way the functions are recorded for each gene is that if a gene is annotated to a term down here, it is also annotated to terms up above. Or in other words, the assignments are transitive up the hierarchy. So there's many, many terms assigned to molecular function, and then fewer terms at lower levels in the hierarchy, which are more specific. Okay, so we can think of this as just a graph problem, a simple bipartite graph where we have terms or functions that are and genes, and they're connected if the gene is annotated to that term. And we want to look at this network for, to understand what information can we gain by analyzing this kind of network. So, because we want to find the relationship between terms, and because on the other hand, we also want to see what the functional similarity implies in terms of relationships between genes, we're interested in making two projections. So we start with this bipartite graph of gene annotations, and we can project onto a term network where we have connections between terms, or we can project onto a gene network. And it's actually this process of how do we actually construct the gene network from this annotation data that is really the difficult one, because there are many things you could do. You could do different kinds of weighting schemes, and in order to find And so we've searched through this and tried to find, well, what kinds of weighting schemes are meaningful in this context. Okay, so once we do these projections, how are we going to use the term in gene networks? So as I mentioned earlier, the term networks are going to be used to group biological functions. So given the kinds of annotations that are submitted to the Gene Ontology, we can say, okay, well, these terms are related through many annotations to many gene annotations. And then we can say, are there groups of functions that are related that we didn't really know were related before? And then on the flip side, the gene networks can be used to understand and predict interactions between genes. Okay, so what is our process for analyzing the structure of the term network? So we're going to think about the term network for a little bit here. So we take our gene term bipartite graph from the annotation files, and we create a projected term network. And I'll tell you about how we do that in just a little bit. But imagine we have a good process for creating a term network. Then what we do is we look at the community structure or the clustering in this network. So we identify clusters of terms that are highly connected within this projected term network. And we want to do some sort of evaluation. Are these actually biologically meaningful groups? Is this some kind of artifact of how the data is, of the way the data is collected? And so what we do is we convert these term communities into sets of genes associated with these communities. And now we can evaluate these sets of genes because there's a separate database of gene signatures. So sets of genes from biological functions which are known to do something in particular. So there's a gene signature for a certain kind of lymphoma, a gene signature for another kind of breast cancer. So we have many of these gene signatures. And if there's a good overlap between our gene communities and gene signatures, then we can say, okay, well, there's something biologically meaningful in the way we've divided up these functions. And for comparison, we want to look at, well, what about the way in which the conceptual organization of the biological term divides up, the conceptual organization of the Go hierarchy divides up functions. So you remember the term hierarchy is human built, right? It's this idea that, okay, biologists think that molecular function is divided into these sub-functions. So it's this human built hierarchy, and we can collapse this into fuzzy term communities. So each community here is then a branch, and all the terms underneath this branch are included here. And so here when I say fuzzy, it's because terms can be included in multiple different branches. And so then we do the same process of finding the genes associated with these branches, and we compare the significance of these kinds of sets to the cancer signatures. To say, okay, well, which view of grouping biological functions together actually is more consistent with the kinds of biological functionings that we find from these gene signatures. Okay, so now for the part of understanding how we actually do the projection. So we can make term and gene networks from a simple projection, but then I'm going to argue that we actually want to do something a little bit more sophisticated. So the Gene Ontology is a bipartite graph. And so if this is my matrix of whether a term is annotated to a gene, I can construct a term network which is contained in a matrix T by multiplying my matrix B times B transpose. And so in that case, the ij element of T reflects the number of genes connecting term I to term J in this network, in the bipartite network. And similarly, I can make my gene network by multiplying B times B transpose times B, and Gij is then the number of terms that connect gene I to gene J. So that's the simple, straightforward way of projecting these networks. So, but then we might want to say, okay, well, maybe we want to weight things down. If a term has a lot of connections, we want to weight down those connections appropriately. Or maybe we want to, if a gene has a lot of connections, we want to weight those down. And so, in order to think about this, one thing we might want to look at is the degree distribution of the terms and the degree distribution of the genes. So this is the number of connections, or the number of annotations to the terms, and this is the number of terms, this is a cumulative distribution. This is the number of terms that have this number of annotations or greater. And so you see, it's a very heavy-tailed distribution. So I should say something about what the different lines are, the different colors are. So black represents all annotations. And as I've mentioned, the Go hierarchy is divided into 3 main branches. And those are biological process, molecular function, and cellular component. So historically, the branches are often investigated individually. And so I'm just showing you the basic properties of the 3 different branches, and they're quite similar, in fact. Now, if we look at the degree distribution of annotated genes, the cumulative degree distribution, we see something a little bit different, that in fact, this is much less heavy-tailed. So we have terms that have 10,000 annotations, but the degree distribution for genes is far more truncated. So yes, there are genes that have large numbers of annotations, but that effect is that the skew is not as strong as the term network. And that's something that when we played around with the weighting, it came to be quite important. So the weighting of the term network. What we start to do, what we did was the following. We want to weight the network according to the degrees of the term. So we want to weight a connection. So if we have a term I and a term J, we can count the number of genes connecting term I and term J, but then we want to weight that down by the product of the degrees, the degree of term I times the degree of term J. And so what this turns out to mean in our formalism is if we construct a diagonal matrix WII, which is 1 over the degree of term I, then our new term matrix is WB transpose W transpose, and has the elements corresponding to what we desire. The number of genes connecting term I and term J divided by the product of their degrees. So what is the consequence of this kind of weighting? How do we then interpret the links? Well, here we see that Tij is going to take on a maximal value of 1 when term I and term J each have, so it should say each only have the same single gene annotation. Tij takes on a minimal value of 0 when term I and term J share no common annotations. And Tij gets small when term I and term J are both of high degree, but they share few common annotations. So once we have this term network, we want to analyze it for community structure. We want to say, well, are there groups of terms that are highly connected through this kind of viewpoint of looking at the connectivity between terms? So we want to identify groups of strongly connected terms. So to do this, one can apply a variety of network community finding techniques. And I've worked in this area for a number of years, and so I'm going to tell you about the techniques that we apply. So the problem of identifying community structure in networks has received a lot of attention in the last decade, and even beforehand, but and a kind of an explosion of papers on this topic in the last decade. So the problem is that given an arbitrary network, we want a method to divide the networks into groups, sometimes called communities, such that within group edges are relatively dense. And one of the important caveats, which makes this a little different than traditional computer science, sort of old computer science defined problems of this nature, is that we do not want to specify the number of groups a priori. Rather, we would like to find a natural division of the network into communities. So if there exists 7 sort of natural communities, we want to find that number rather than saying, okay, well, what's the best way to divide this network into 7 communities? And so here is just a graph that shows you strong community structure, or sometimes we call it modularity. So here you see this is a friendship network, and it's divided into groups such that within group connections are quite dense compared to between group connections. Okay, so in order to detect the community structure, it's useful first to develop a metric to say, okay, if I have a partition, is that a good partition? And so this is work that I did quite a long time ago when I was a postdoc, actually maybe when I was a grad student, on quantifying the community structure. So we can calculate the strength of a given partition of a network into k communities. The strength of a given partition of a network into k communities can be quantified by something we call the modularity function. And it's defined as follows: the sum over all communities of EI over M minus DI over 2M, the quantity squared. And so what does this mean? So here, EI is the number of edges that connect vertices in community I. DI is the number of edge ends that connect two vertices in community I, and M is the total number of edges. So basically, what this is measuring is the observed within community density versus the expected within community density. So you take a given partition, you apply this measure, and you say, is it a good partition of the network into that number of communities? But as I said, our goal is to identify a partition not specifying the number of communities a priori. And so this can be done through modularity maximization. So basically, the problem is to find the partition that maximizes this modularity function. And this is proven to be an NP-hard problem, but many heuristics work very well in practice, although they have several caveats. So the heuristics that work well in practice are just greedy agglomeration, different kinds of spectral methods, and simulated annealing. I'd like to just mention a few caveats because I'm going to go through using this kind of approach, but saying, well, really we can analyze this data with any kind of network clustering tools. So this approach basically, it's a strict partitioning of the network such that if a term gets put in one community, it's not also placed in any other community. And also, it tends to, a lot of times in networks, there's a degeneracy in the optimization. And so that's something that you might want to explore, and exploring that degeneracy can tell you about how, well, maybe these terms should be put into multiple different overlapping communities. And that's been a topic of some other work that I've done, actually in collaboration with Carl Kingsford, whom some of you may know. And one last caveat to this process is there tends to be a resolution limit problem to this approach of network clustering, that you can't find clusters smaller than a certain scale. And so if you're interested in very small clusters of functions in our gene annotation problem, you actually have to do a little bit of adjustment to your community finding process in order to find very small clusters in order to fix the resolution problem. Okay, so back to the term network. So I take my term network that I've built from my projection of my bipartite graph, which has been weighted in the appropriate way, and now what I do is I divide it into communities based on the process that I just shared with you about modularity maximization. So I get some number of communities, and let's say, it's around 15 in this case. The best division of the network into communities is about 15. And now I'm taking, this is the entire Go hierarchy for one branch, molecular function, I believe, although I can't see it here. And now I'm coloring the functions or terms based on which community I found them to be associated with. So what we expected a priori was that we would get color, maybe we get dark blue over here and cyan over here, and maybe some mixing. But in fact, what you get from this approach is that the communities are completely different than the groupings of terms in the hierarchy. So we get out that if you group terms or functions by the annotation data, you get a completely different view of how functions are organized as compared to the hierarchical view, which is that predefined conception of how functions are related.
How did you come up with such a hierarchy?
So the hierarchy is predefined by the Gene Ontology itself. So I didn't build this. So a consortium of biologists built this hierarchy. And so from a variety of, by combining a variety of different results on how functions should be related. And so our basic question is saying, if I find clusters from the annotation data, do they divide in ways that are similar to how the functions are divided in the conceptualization in the hierarchy? Does that answer your question?
Yes.
So can I ask?
Yes.
So, just sort of trying to wrap my brain around this. Terms would end up in the same community if they share a lot of genes in common or something. Like
Yes.
if those functions sort of have the same genes.
Yes, they share a lot of annotations, but we weight down, but then we weight by the degree of the term. Yeah. So it's like a statistically significant number of annotations that they share. Exactly. Okay, so this is just a close-up. And you see you get some things within a branch. So this is a lower level branch, and now I'm coloring it by communities. And you have some kind of enrichment between the communities and the branches. So here we have this term, and now we look at, okay, well, this lighter color here, this aqua color, there's a few terms from that community that are in this branch, but basically we see all kinds of different colors and not a very strong overlap between the functional sets we find versus the functional sets that are predefined. And I should say that because of the way the annotations are defined, we expected to see more, right? Because each gene, if it's annotated to here, it's also annotated to here and here and here. So we didn't, it's not that we only kept the annotations to the lowest term to the lowest leaf. We kept all the annotations all the way up the hierarchy.
So what's the biological meaning of that?
So what's the of the kinds of,
The fact that, for example, the one at the bottom that you indicated, I don't know, what's the name of that color in English?
purple or?
Purple, yeah.
Sure.
The fact that purple is not directly connected to the other purples, for example.
So the biological meaning to what we see here is that if you look at the annotation data and you say, well, what are the functions that genes are connected to through this annotation data, through several different experiments, kind of from a agnostic point of view, you see something very different than the conceptualization, which could imply that actually there are stronger ties between functions that aren't connected in the conceptualization. It's not that there are no ties between these two kinds of functions. It's just that the ties between other functions tend to be stronger in our viewpoint. And this could be a partial artifact of the kind of annotations that are submitted. So, you could say, okay, well, annotations are basically usually submitted based on certain types of experimental data, and now we're biased to find those kinds of connections.
So the fact that you had, for example, this example has led to a revision at the biological level as to what people thought was happening?
I wouldn't say it's led to a revision of the biological level, but it's leading to potentially an alternative way of evaluating, a set of genes to say, okay, what functions are they associated with? If you group functions according to our way, you can find, okay, well, now they're associated with these kinds of functions, versus if you're just restricting yourself to the hierarchy, you might have a gene set that is related by function, but it's all over the place on the hierarchy. So it's a different way of saying, okay, can we classify the function of the gene set by learning about the relationships between functions in this way? You had a question?
Yeah. Were you concerned at all that your degree weighting scheme was causing this, that you were kind of throwing out the high degree genes?
Yes, we were concerned, and in fact, we tried numerous other weighting schemes. But the way in which we started to believe that, even though, okay, so basically you could do something arbitrary you do many different things to create this term network. And some of those things would create things that weren't would create term groups that weren't biologically meaningful at all. So our validation of the kind of approach we used came in separately investigating the validity of our functional groups, which is what I'm going to get to next. Okay, so in order to investigate, so as I said, we could, you can do a term projection, you can find communities, but the question is, do we find anything biologically meaningful? And that's our goal in this next step here. So what we want to do is compare the biological significance of the communities of terms that we find through this term projection network clustering process as compared to the branches of this hierarchy.
Sorry, before you move on.
Yes.
If you throw different community detection algorithms, you always get similar pictures?
We get similar pictures, although we haven't explored that very extensively. But that's a very important point, because we are, having worked in this from this modularity direction, that tends to be the kind of thing that I use. And we've done a little bit different clustering, but I can't really say. I think that's an important point that we should investigate further. Okay, so we take these network clusters and we compare it to the groups that are given by the different branches. What we can do is we can convert each of our communities into a group of genes. And we can do the same for the branches. And now we want to say, if we look at gene sets that are experimentally validated to be associated with different kinds of biological processes, such as, as I said, gene signatures for cancers, and heat shock and things like that, now we divide these. So the question is, do we find in our communities significant overlaps with these kinds of externally validated gene sets for different functions? So we take a compendium of gene signatures from a cancer database, and then we just calculate a hypergeometric probability for our sets and as compared to the signatures. And in fact, the hypergeometric probability is kind of a bad statistic to use here. We've actually developed a little bit better statistics for this kind of comparison, but I'm just going to talk about the simpler one here today. And so, what do we do? Okay, so if I take my different cancer signatures, and here's a list of the different cancer signatures, and here are different communities that are created by the branches, and these are the communities that we've found. And now I'm saying, okay, well, what's the enrichment of these different cancer signatures across different communities in different branches. And what is the statistical overlap? And so darker colors represent high statistical overlap. And you see that actually we tend to have a bit more statistical significance with these biological gene sets than the terms, the branches. And then in addition, you see that you get different information. Because our communities don't really overlap very well with these term branches, you can get, for instance, let's say this gene signature here is associated with this function and this branch over here, but it's also very strongly associated with a different grouping of functions. So it's a new way to get different information for how to group these functions and interpret a set of genes in terms of, say, okay, they do a certain kind of function, they're associated with a set of functions, but it wasn't known a priori what that set of functions was until you do this kind of analysis on the annotation data. Okay, I want to switch gears for a little bit and talk about the implications of functional similarity for gene regulatory interactions. So far I've talked about, well, what can we learn by analyzing the term network? And now I want to talk about what can we learn by analyzing the gene-gene network. So the question is, why do we want to make a gene network from gene annotations? Well, it's a really cheap and easy way to generate a gene network that is species specific, for which there's no or limited experimental gene networks. And the experimental gene networks I'm interested in are gene regulatory networks. So in order to derive an experimental gene regulatory network, it takes a lot of different experiments, and you want to run a lot of high throughput experiments together. And so, and a gene regulatory network, I should say, tells you if gene A regulates or the production of gene B's protein, right? And so this is an easy way to generate a gene interaction network that takes into account disparate experimental information. And in addition, we can use this to interpret known gene regulatory networks. So we can look at, okay, well, what's the functional similarity of genes in known gene regulatory networks? And also, it can be used to evaluate and or improve existing network reconstruction algorithms, because this is a cheap way to take functional information and try to infer gene regulatory interactions. So that's our basic goal. So what are we trying to do here? So we take the bipartite graph, and we want to calculate an appropriate projected gene network. And we also want to say, okay, what information does this give us about true regulatory interactions? So we take an experimental gene network that's validated separately, and then we take another kind of network that's reconstructed from microarray data. And so this is a well-established, this is sort of our gold standard network. This is the high throughput way of finding interactions. And this is just the annotation network data of saying, okay, can we actually find any interactions through this? And we kind of compare these different things and potentially develop an improved method of reconstruction that uses the annotation data in addition to the microarray data, the high throughput data. Okay, so again, one of the things we have to deal with is what's the appropriate way to weight the gene network? So since I weighted the term network by the degree of terms, you might think I'll weight the gene network by the degree of genes. And in fact, that was the first thing we tried. But in terms of identifying the ability of our projected network to predict edges in our gold standard, we actually found that weighting by the degree of the terms was most important. And in fact, we didn't do just a straightforward weighting, but we weighted to a power alpha. So our weight matrix, Wij, was 1 over the degree of term I to the alpha. and I'll talk a little bit about what the implications of this alpha are. So, in the limit of large alpha, edges in our gene-gene interaction graph take a particular ordering such that those genes connected through many low-degree terms have the highest weights. So as alpha gets higher, then it's the low-degree terms that become most important. So to be a little more specific, the consequences of weighting G with large alpha are the following. Gij is largest when gene I and gene J are connected through many low-degree terms. Gij takes on a minimal value of 0 when gene I and Gj share no common annotations, and also that Gij gets small when gene I and gene A are only connected through a single high-degree term. So the reason we have this alpha parameter is we want to understand, well, what kind of gene network can you extract from the gene annotation data that actually tells you about real regulatory interactions? And so that's why we sort of started with a large, we explored many ways of weighting the data. Okay, so how do we compare this to experimental data? Well, we construct our matrix G, and then we apply a threshold to this gene-gene network that we've created from the annotation data, such that every gene pair whose Gij is above the threshold is considered connected. And then we compare this derived network for a certain threshold to the gold standard regulatory network. And we do this for E. coli. And then for each threshold, we calculate what we call the F-score, or what is called the F-score, to measure the utility of our gene-gene network for capturing true regulatory interaction. So the F-score basically measures the trade-off between precision and recall. So precision is the number of true positives over the total number of positives, and recall is the number of true positives over the total of the number of true positives plus false negatives. So basically, you want your precision and recall to be high, so if you want to combine these both into a single statistic, we sometimes use the F-score. And so the maximum power of our of the gene-gene interaction network is going to occur at the threshold value where the F-score is maximized. However, we don't know externally, so that's sort of the maximum inference power of this approach, but I'm going to tune that to its maximum, but we don't have a good heuristic for if you didn't have this gold standard, how would you tune that? So that's a caveat to bear in mind. But if we look at the inference power of this method as a function of this weighting parameter alpha, you see that we get better and better as alpha gets higher, and then then it plateaus. And so the implication then here is that really you want to make the low degree terms as important as possible, and if you make them maximally important, then you get the most information out in terms of inferring these regulatory interactions. Okay, so what we wanted to do is we want to compare this very simple approach to high throughput data approaches. So our simple approach just takes the annotation data and tries to construct regulatory interactions. It tries to infer regulatory interactions, but you could also do a really expensive experiment perturbing the genes in the network and looking at the gene expression over many different experiments and looking for correlations between genes that go on and off together. And so there's a method that we compare to, which is widely cited, called context likelihood of relatedness. And it calculates the mutual information between pairs of genes using this expression data. And it applies a z-score metric to evaluate the statistical significance of this mutual information. And it turns out that neither of these methods is very good, but they do give you some information. So now, if I say everything above a certain cutoff, z-score cutoff, is going to be in my inferred network, or everything above a certain scaled similarity is going to be considered in my network, you can see that if I look at the F-score as I vary the threshold for the Z-score, or as I vary the threshold for our similarity measure, which we call our scaled similarity, then you see you get different regions where you have high significance, a higher inference power. So your Z-score, at high Z-score, I have higher inference power, at high scale similarity, and these are very different sets of edges. Actually, there's not a lot of overlap between what we infer and what is inferred through the high throughput procedure. So what this means is that you can improve network reconstruction algorithms by simply doing a combination of these two approaches. And I should mention that people have incorporated Go data in their network reconstruction algorithms, but really mostly as a means of looking at individual pairs and trying to say, do we want to validate this pair or not? Not as trying to infer, just as, not as, basically as a way of fine-tuning the interactions that come out of high throughput data. But what we find is it's actually better as a way of finding alternative interactions that don't come out of high throughput data. So it's not that people haven't used the annotations to improve network reconstruction algorithms, but the way in which they've used it is just to refine the set of edges that come out of the high throughput. But in fact, if you use it to find an alternative set of edges, you do better. So if we look at the percentage of edges in the inference method that are actually in the gold standard as a function of the threshold, then we see that we're doing about the same, actually, on our projected network as the high throughput method. I mean, we do a little worse, but actually not significantly worse. Our method is completely cheap versus doing these large experiments, which tells you if there's a species that is well annotated, but it doesn't have good experimental data, you can get a lot of information about regulatory interactions this way. But then if you combine them, because the sets are so disparate, then you can actually gain information just by combining the two. Okay, one thing I want to mention is that there are other measures of functional similarity. So we've used the network, we've used a certain method for projecting our annotation data into a gene-gene network data, which gives it into a gene-gene network, which gives us a certain measure of the functional similarity, right, between pairs of genes. But there are other traditional measures of functional similarity, and I want to compare the inference power of those methods, but I should say that they're not designed to infer regulatory interactions. So what we were trying to do in our weighting scheme was try to find, well, what is a good weighting scheme for inferring regulatory interactions? And the other kinds of metrics have different goals. But if you just compare different similarity methods, measures, these are two, kappa statistic and semantic similarity, and you look at, well, what's the maximum predictability in terms of predicting the edges in the gold standard, that we do pretty well here, and in fact the best of this set if you take this SA, which is where alpha is large. Now if we didn't do the alpha weighting for comparison, we took S0, then you see that our predictive power is significantly less. So that just motivates why we do the alpha weighting. Okay, and then finally, having constructed this measure, because we find different kinds of edges that are found in the high throughput data, we might ask, well, what kinds of edges do we tend to find that high throughput data does not? So what does it mean structurally to have high functional similarity? So to measure how structurally important or redundant an edge is in our gene-gene graph, we calculated the new shortest path between the nodes. So we took an edge here that we say is connected, And then we looked at the experimental network, the gold standard, and we said, okay, well, how important is that edge in the gold standard network? And we do this by calculating the new shortest path between these two nodes upon removal of this edge. And so if the new shortest path gets much, much larger, then that edge is really important for connecting those nodes. And so here we're plotting the harmonic mean of the new shortest path as a function of the index of the edge in the gold standard ordered by edge weight. So high edge weight edges that are also in the gold standard are really structurally important. So we've come up with a measure that really correlates well to this kind of structural importance. And if you see, we do the same thing on the high throughput data. The edges that are important according to their high throughput scores don't tend to have this feature of being structurally important in the gold standard. Okay, so just to conclude, what have we done here? Basically, we've tried to mine the gene annotation data using network techniques. And we found that there's an alternative natural way to group Go terms, unique from the hierarchy, and it provides an independent framework with which to describe and predict the functions of experimentally identified groups of genes. And we also found that the gene ontology and annotations to the gene ontology can be used to create a gene network entirely based on functional annotations. And properties of this network are known to correlate with known regulatory interactions almost as strongly as very expensive high-throughput experiments. So this gene network also, it identifies a different subset of regulatory interactions than those provided by the high-throughput approach, and can be combined with high-throughput approaches to further improve predictive power. So with that, I'll stop and thank you for your time, and I'll take any questions.
Speaker, before we open the floor for questions, let me remind you that we have an open discussion roundtable at 3 o'clock in 2168. Anybody who wants to join in can join in. And let me open the floor for questions.
When you construct such graphs, how do you in general validate? Because it seems like the kind of the data set you can validate is fairly limited. So, I mean, for one set of data it may be good, but if you actually deploy it.
Yes, I mean, it's quite a challenge, right? So on the gene side, it's a little easier because there are separate data sets for regulatory networks. And we did our analysis for E. coli, where there's really quite a good data set. You could also do it for yeast, there's some data sets, but then for human, it's not very good at all. So it's a big challenge. And so we've done it on just E. coli and yeast, but then to say that, okay, well, it's going to work just as well on other kinds of other species, it's a bit of a jump. But my hope is sort of to continue our validation procedure as more and more data comes out. So there is data in human, it's just not as good. We can't really consider it a gold standard. And then on the term side, it's actually even more challenging a problem to say, okay, well, what's the meaning of these groups? And we only do it by saying that these term groups are just as meaningful in terms of experimentally known sets of genes as the branches. But it is a challenge.
Any question? I want to thank you. At the beginning of your talk, you took the B matrix, which basically is the adjacency matrix, the bipartite.
Yeah.
Then you took the B transpose and then B transpose B.
Yeah.
You, if I understood correctly, then you should be getting blocks that are all zeros, right? Because aren't you trying to? I mean, it wasn't the rule the fact that two blue nodes were connected if and only if each had connected to the same, at least one red node,
Right.
which was the identical.
Right.
So when I take BB transpose, somehow I must get for the blue and the red in this new matrix, I should get the term to be 0 in that case, right?
You should get what to be 0?
0, the
What to be 0, sorry?
BB transpose, the corresponding IJ entry for BB
Oh,
transpose.
right.
That's what
Yes,
I get,
yes,
right?
yeah.
Any other question? No? All right, let's thank the speaker. Thank you.